So I think we're going to start. It's a great pleasure to welcome you all. This is the last of our 6 lectures in our distinguished lecture series that's underwritten by AARP, and it's celebrating their 50th anniversary. And as I've said in previous lectures, that they chose us as a venue to highlight some of the major contributions in gerontology that people have made and are making today. And it's a great pleasure to thank them once again for their support and continuing interest in our school and center with whom we share a long and productive history. And I've just managed, did I do that? Or I thought I hit something. That's our mission statement, a bit wordy, but it's sort of what we're about here at the Davis School. We're an all-purpose research and educational enterprise here at USC, one of the things that USC is well known for, and we're very proud to be part of that legacy and part of the present and future of this fine university. Our speaker today is Rudy Tanzi, who holds the Joseph and Rose Kennedy Chair in Neurology at Harvard Medical School. Dr. Tanzi received his bachelor's degree from the University of Rochester in history and microbiology. I guess he chose one path and not the other in terms of his life's work. He went on to earn his Ph.D. in neurobiology at Harvard in 1989 and has been on the faculty of Harvard Medical School for some time, I think pretty much since then. One thing I only learned about him recently is that he, among his many accomplishments, he's a jazz pianist, and he just mentioned to me that he sometimes, what do you musicians call it? Not rumble, but jam. I don't know if you rumble too. I don't know. Rumble too. He jams with our own Tuck Finch, and who many of you may or may not know that Tuck is also an accomplished musician. I think he's on the fiddle or the violin. I'm not sure what he calls it, but he plays. Anyhow, it's a great pleasure. You have a bio about Dr. Tanzi that was handed to you, so I won't go over most of what's there. He's very well known in biological and genetic research in various diseases, in particular Alzheimer's. He is scientific co-founder of a firm that's unlike what you have in your bio. It's called Prana Biotechnology, and of particular note, he are 2 prizes which he is the recipient of for research in Alzheimer's. One is the Metropolitan Life Foundation Award for Medical Research, and the other is the Potamkin Prize for Research in PICS, Alzheimer's, and related disorders, which he won at the advanced age of 36 in 1995, and is known in many circles as the Nobel of Alzheimer's research. So he's accomplished a great deal, and is still in this context, Rudy, you're a very young person, so it's one of the nice things. It's cool to be an older person here in this building, and it's nice to have young people come in like yourself of such great distinction. We're very, very happy to have him here, welcome him, and look forward to his remarks. Dr. Tanzi.
Probably should have put this on before, but I forgot. Thanks very much to the Dean for a wonderful introduction. I always know when they're good if they make me blush. And thanks for the kind invitation to be here and talk to you today about Alzheimer's disease. And it's always wonderful to come back to USC and give a talk. Come back every few years. And it's great to see good friends here, like Tuck Finch, the old fiddler. If you want to hear something strange, bluegrass fiddle with jazz piano, but somehow it works. And if we had it here, we would play right now. And Ed Schneider, who I've known also for years, and I feel kind of like a bit of a reunion whenever I come back here to give a talk. So thanks for coming. And I did originally prepare a talk that was very scientific, and then I realized that much of the audience will be a lay audience. And so since I'm not here to put you to sleep, I changed my talk around at the last minute so that hopefully there'll be enough science for the students and the scientists in the group, and hopefully enough general information for the lay audience, and we'll see if this works. I urge you to ask me questions during the talk. Keep it informal. And I may ask you questions, so you have to, and I may point to you and ask a question. No, I won't do that. I'm just kidding. But so we'll keep this casual, and I'll try to learn something about Alzheimer's disease. For conflict of interest, there are a number of companies I'm involved with. I'm co-founder of 2 companies. One's still doing Alzheimer's research, Prana Biotechnology, and I'm a consultant for a number of companies, AZI, Siemens, Neuroptics, Pathway Genomics. And these days, it's just important to be safe and show these types of conflicts up front, because you never know when I might mention something from something one of these companies are doing. So what I'll do in my talk today, and I should say that the first time I spoke at USC, I think it was in early 90s, I got done with my talk, and Ed Schneider came up to me, and he said, "Rudy, you know, you have a lab now, you're a young PI now, and when you give a talk, you really need to tell them first what you're going to tell them, you know, so that they know what to expect, and then give the talk, and then summarize." And ever since then, Ed comes into my head. It's terrible. Every time I give a talk, which is quite often, Ed comes into my head and says, "Now remember, tell them what you're going to tell them first." So I thank you. Thanks for that, Ed. So what I'll be talking about today is first an introduction in general to Alzheimer's disease and what is it, what do we know. Then I'll talk about the genes we've discovered over the last 25 years and how these genes have taught us really more about the disease than we ever knew. And the genes really changed our whole view of Alzheimer's. And then a little bit about what the genes taught us biologically. And then the point that these aren't all the genes. We've had a great ride on the genes we've discovered, but there are more genes to find. So I'll talk about the Alzheimer's Genome Project and the search for novel Alzheimer genes. And then finally, what have we learned from these genes so far in terms of emerging therapies to really treat the disease itself and not just treat the symptoms like current drugs do. So in terms of introduction, as you know, Alzheimer's is the most common form of dementia in the elderly. Often it's diagnosed as dementia because a confirmed diagnosis requires seeing certain pathology in the brain to say, yes, this is dementia due to Alzheimer's. But Alzheimer's is a type of dementia, and the most common you see in the elderly. And like I said, most cases are termed either dementia or probable Alzheimer's disease, but the vast majority of dementia in the elderly turns out to be Alzheimer's upon autopsy. Risk factors include age is number one. Prevalence increases dramatically with every decade after 60 years old, finally getting to up to 40 or 45% of the population after 85 years old, being at least in the early stages of Alzheimer's, so highly prevalent disease. Family history is what got my attention. This is a genetic disease. One of the most seminal studies came from Dr. Gatz here in collaboration with a group in Sweden showing in studies of twins that up to 80% of cases involve inheritance. And I believe that at the end of the day, it'll be tough to find a single case of Alzheimer's where some gene, no matter how minor its effect, is involved with the disease, that genetics plays a complex role, in that you have familial cases where you have usually early onset genes involved that I'll talk about. But I want to make the point that when we say sporadic Alzheimer's disease, that doesn't mean non-genetic. And this is really something you see often where people will say, well, the familial cases are rare and those are genetic. The rest is sporadic and not genetic. Fact is, genetics affects both the familial cases, where you see at least 2 first-degree relatives with the disease, and in the sporadic cases, often later onset, where you may not see familial clustering, you still see a very strong genetic influence. So we need to find all of the genes involved. Some life exposure factors include head trauma and stroke. This is getting a lot of attention these days. Obesity, high cholesterol levels, and I have included insufficient federal funding as a major risk factor. And we're really hoping that this will change. with the current administration, but we're fighting. I and others heading toward Washington every few weeks and going to Capitol Hill and the White House a couple of weeks ago and really arguing that, the funding for Alzheimer's is dismally low, just unacceptably low in this country. When you look at how many cases we expect to see between now and 2030, this disease isn't going to go away. And it's believed that somewhere in the late teens, if we're where we are now, we're treating this disease, that's the tipping point, that we will see a major breakdown of Medicare and Medicaid, given the number of Alzheimer patients we have in this country by the mid-teens, if we're at the same exact state we are right now in trying to treat and prevent this disease. This is $470 billion. But yeah, I mean, these costs, it could be even higher. I mean, it's hard to say. The point is just the exponential increase if we don't do something. And already, Alzheimer patients make up 12 or 13% of Medicare and Medicaid, and they suck up 1/3 of all of the costs. And that's now, and that's at this point here. So you can imagine what's going to happen as we head over the next decades. The federal funding for Alzheimer's looks like this. This was in 2003, $723 million, and this is what's happened over the last few years. And if you go to 2008, it also went down. So asleep at the wheel comes to mind. Basically, real problem as the prevalence of this disease goes up, funding continues to go down. And I also heard in Washington a couple of weeks ago that even though the funding is supposed to be now at the 600+ million level, federal funding, that when you really look at the books and do the math, it turns out it was probably about 400-something million. I'm not sure what the discrepancy was, but this is just something that was going around Capitol Hill about 2 weeks ago. So we just need a whole lot more money if we're going to keep up with this disease and not let it really just strangle the healthcare system in this country. So let's get to the science then. Alzheimer's is named for this gentleman, Dr. Alzheimer, and in 1906 he studied a female patient who had been admitted to the asylum there in Tübingen, Germany in 1901. She complained of delirium and well, I think her husband did this, complained of delirium and frenzied jealousy toward her husband. Probably he wrote those notes. And anyway, she was suffering from dementia, and upon autopsy, it was Alzheimer who saw that her nerve cells and neurons were choked with these dense fibrils, and he saw that outside the nerve cells there were these big bundles, plaques he called miliary foci, and he had the bold option at that time to say this was causing her dementia, these physical lesions. And he was giving this talk in Bavaria. Carl Jung was in the audience. It was probably pretty intimidating. And his talk went over very badly because at that time it was so difficult for people to think of physical lesions causing a mental psychiatric illness. And so it was very revolutionary for him at that time to do this. And for a long time, cases of pre-senile dementia, in other words, dementia that strikes under 60 years old, was called Alzheimer's disease. But it was limited. Alzheimer's disease for decades was limited to just these pre-senile cases where onset was under 60, like in the original patient, August D. And it wasn't until the 60s that it was seen that this same pathology Alzheimer described, which we now call senile plaques as the miliary foci, his fibrils we now call neurofibrillary tangles. The plaque has a center made of the sticky material called beta amyloid. You also see it around the blood vessels in the brain. And this pathology was so frequent in cases of senility that we realized that in the 60s and 70s that this is mostly Alzheimer's disease. So suddenly the prevalence of Alzheimer's went up dramatically because it wasn't just reserved for these early onset cases. It was most cases of age-related senility. And the next big breakthrough came in the 80s when George Glenner at UCSD, who studied the sticky material called amyloid, he proposed that Alzheimer's was an amyloidosis of the brain. Now, amyloid is an old term from Virchow from the 1800s, and he was studying plant starch, and it meant starch-like amyloid. And but But it's all over the body. There's all different types of amyloid, in the heart and the spleen. We get it as we age. And different proteins become abnormally manipulated to make amyloids. And in Alzheimer's, Glenner was saying back then that, well, this is yet another amyloidosis that comes with aging, but it's in the brain. And we have to figure out what the amyloid's made of. And in '84, he published a paper saying that the main component of the amyloid in the blood vessels and in the plaques of Alzheimer's is made up of a little protein we now call A-beta. He originally called it the amyloid beta protein based on his lab notes and terminology. So A-beta now existed, and at that time I was a student at Harvard Medical School, and I decided to go after the gene that makes this material. Other groups around the world were doing the same at the mid-'80s. And in '87, our group and two other groups in February of '87 published the gene that makes the amyloid beta protein. And I had the privilege of naming this thing, not very creatively, the amyloid precursor protein, because the gene made the protein that was the precursor of the amyloid. So not the greatest name, but we call it APP for short. And since then, this molecule has been the major target of drug discovery in Alzheimer's. And I think most would argue that before we found this gene, we really didn't know what to target for therapies and drugs. But this now gave us a protein, a gene to target for drug discovery and drug development. And what we learned was APP sits in the cell. This is the cell. Here's the surface of the cell. It sits so that you have an extracellular portion here. This is going into the membrane of the cell surface, and then this part's inside the cell. And then we soon learned that you had to get two clips. First, a clip here by an enzyme we call beta secretase, and then a second clip here by an enzyme we call gamma secretase, and that releases this little piece of the protein that we call A-beta. And if you get lots of A-betas made they in the brain, they start to stick together, and then eventually they form a plaque. So this was a big revelation for us in '87, that the junk in the plaque wasn't just junk. It actually came from a precursor protein. And of course, drug companies said, well, we can target these clips, or we can target this accumulation as a way now to treat the disease. If we try to block this stuff from accumulating, we can treat Alzheimer's this way. And to this day, this is now the most promising set of therapies that we have waiting in the pipeline. It wasn't until a few years later that the first actual mutations were found in APP. The first one was actually found in a Dutch family that had just blood vessel amyloid causing hemorrhage at a very early age in the 50s. And then right after that, a mutation was found that caused early onset familial Alzheimer's disease. And this was an English family from London, so it's called a London mutation. And what was interesting is that these mutations hover in APP right around the membrane where the A-beta is. So here's that, like I said, here's that first clip. There's the second clip, and it releases the A-beta, and you see the mutations hover right around these points. This clip in the middle is a good clip that gets rid of the A-beta. That's what we call the normal clip. But when you get this one and this one, you release the A-beta, and now we know of all the different mutations all hover in the same region. And what they do is they drive up, this is an important point, the ratio of A-beta 42 to A-beta 40. And what that means is that if you get this first clip at position 1 of A-beta here, and then you come down A-beta, and you get the second clip down here, that second clip can either produce a peptide that has 40 of these amino acids or 42. It's the longer version with 2 extra amino acids here that is most dangerous, because it's the A-beta 42 that promotes the aggregation of A-beta into these toxic assemblies that eventually will turn into plaques. So what we've learned is it's not so much about how much A-beta you make in the brain as we age that will determine your risk for Alzheimer's, but more importantly, the type of A-beta you make. So normally, 90% of the A-beta you make is A-beta 40, about 10% is A-beta 42. But if you tweak that a little bit, so let's say instead of 90-10, it's 88-12, that's enough to skew the ratio, and now you start driving the production of A-beta aggregates. So this is demonstrated here. The single A-beta, the single molecule, is pretty safe. It doesn't seem to cause any problems. But as soon as it starts to bind together, where you make what are called oligomers, that means you can have 2 stuck together as a dimer, 3 of them stuck together as a trimer. These oligomers can lodge themselves in synapses where neurons are communicating and impair neurotransmission, which of course is the basis of cognition, learning, and memory. So I like to say that these little oligomers can short circuit the communication in your neurocircuitry in the brain. You know, this is a slide from my book, Decoding Darkness, and back when I published a book in 2000, we had the little pieces going directly to these fibrils. We didn't even have the oligomers here, and it looks like these are probably most important. We've only been really studying those in depth over the last few years. And I should also say that Tuck Finch and Grant Kraft were really the first ones to study what we would call the first oligomers. They call them Als. You'll see a theme here that Tuck Finch is usually about 10 years ahead of the curve in Alzheimer's. I don't say it just to make him feel good, even though he's a good buddy of mine. He plays a great fiddle. But he usually is. So we had dinner last night, and he was telling me all new stuff, and I had to keep thinking to myself, okay, remember this because it's going to, about 10 years from now, will be obvious. So that's Tuck. So anyway, the oligomers go on to form these protofibrils and these big fibrils, and none of these are really safe. These impair synaptic activity. These fibrils here, you have 5 of them making a pentamer, forming a hollow tube. They can disrupt the neuron and create pores and make them leaky, and they die. And then even when you get these big spaghetti-like fibrils that are in the plaque, they can cause inflammation and gliosis, which can also cause more neuronal cell death. So everything past the single peptide can start to cause problems at different levels in the brain. I should also mention, just to be complete, Brad Hyman in our department recently found that the plaques can also cause calcium dyshomeostasis in nerve cells and cause cell death that way as well. So, but much of the focus these days is on synapses. We have billions of neurons in our brain, trillions of synapses. This is a process from a neuron connecting to another one, creating a synapse. And it's these A-beta oligomers, dimers, trimers, hexamers that lodge here and disrupt neurotransmission. And much of the focus in the field now is on this particular assembly of A-beta as the toxic entity in Alzheimer's disease. In '95, we, together with Peter St. George Hislop and Jerry Schellenberg, found the next early onset Alzheimer's genes. We named them presenilin 1 and 2 for presenility. And it was interesting because the presenilins fit right in. They're necessary for that second clip. So I told you that APP, you get the first clip by beta secretase, second clip by gamma secretase. That releases the A-beta from the APP. The presenilins are required for that second clip, the gamma secretase clip. And there are many mutations in the 2 presenilin 1 and presenilin 2 genes. And again, we see the same thing. The same way the mutations in APP, which is cleaved to make A-beta, drive up the 42 to 40 ratio. Most of the mutations, all but a handful of about 170 mutations in presenilins, also drive up the ratio of A-beta 42 to A-beta 40. So, you know, in Alzheimer's, we tend to be a little dense, but you slap us in the head enough with some information, we finally get it. It's all about the ratio. It's not just how much A-beta. And this is important because for many, many years, biotech, pharmaceutical companies were targeting APP to lower A-beta levels. And that's not what it's about. It's about affecting this ratio of A-beta 42 to 40. Same way, think about cholesterol. You can have lots of LDL, bad cholesterol. But if you have a lot of HDL, good cholesterol, that can offset it. Same thing here. You can have an animal model with lots of A-beta 42. They're getting pathology and plaques. You flood their brain with A-beta 40, and you can actually help them. So it's the ratio that matters more than the amount from the most recent literature. Any questions so far? Any? Yeah.
How do you detect the amount of each?
The way we detect the amount of each is, you know, it's hard to detect it in the brain, of course, in a living person, unless you do a biopsy, and we like to say you need that like a hole in the head. I love that joke. Thanks for letting me tell it. But we can track A-beta levels in the blood and plasma and in the cerebrospinal fluid by lumbar puncture. And there are what are called ELISA antibody assays where you can measure 42 and A beta 40. So far, though, correlating the levels of 42 and 40 in the plasma and the cerebrospinal fluid to the brain have not been easy. We're still working out those details in terms of using that as a biomarker is still being worked out.
Are you able to find out just about when that changes in people?
Yeah, so it looks like as the disease first starts, and you look in the cerebrospinal fluid as a reflection of what's happening in the brain, the 42 starts going up relative to 40. But then as the disease takes place and progresses, the 42, A beta 42 levels go down in the cerebrospinal fluid. We think because there's so much plaque and so many aggregates that it's sucking up the A beta 42 and not letting it out of the brain into the cerebrospinal fluid. So first it goes up and then it comes down. But until we know the exact profile of that relative to the progress of the disease, it's tough to use it as a diagnostic or a biomarker, and that's what people are working on now. Yeah?
Just save the yes for later, just a yes or no. This looks like it's amenable to reductionist biochemistry with the multimer formation after the cleavage reaction.
Yeah.
Biochemistry on it?
Yeah, I'll get to that. Yes, that's exactly where I want to go with the therapy slide. Thanks. Okay, so what we know so far about the early onset genes, there are 3 of them. There are 29 different mutations, actually 30 now, in APP. There was a new one published last week. About 177 now in presenilin 1 and 11 in presenilin 2. So over 200 different mutations spread across these 3 different genes, chromosome 21, 14, and 1. Any one of these mutations is devastating. When it's inherited, it guarantees the disease with 100% certainty. So we say that these mutations are fully penetrant. They guarantee early onset Alzheimer's disease, usually striking under 60. And in fact, the presenilin mutations, which are the bulk of them, presenilin 1, the average age of onset is 44 years old. Luckily, these terribly hard-hitting mutations are rare. They're only 2% of Alzheimer's, and they're mainly restricted to the early onset familial forms. But most of what we're doing in academia and what's being done in industry for drug discovery, drug development, understanding the cause of the disease, most of that information is coming from understanding exactly how these mutations cause the disease. And as I said, the vast majority of these mutations drive up the A beta 42 to A beta 40 ratio. Just to be complete, what the other mutations do, there are a handful that sit right within the A beta region of APP here. See, most of them are down here where the cut occurs, and that affects the 42 to 40 ratio. But some of the mutations within the A beta domain itself can affect its rate of aggregation. So in other words, when the A beta is made, it might form those aggregates, those oligomers, and the toxic assemblies faster. So that's for a handful of them. Moving on to late onset disease, it's a bit of a different story. So as you probably could imagine, I mean, the majority of Alzheimer's disease is late onset. It occurs after 60 years old. We use 60 years as a cutoff. And for late onset, instead of seeing these really rare, hard-hitting mutations that guarantee the disease, we see a different genetic profile. We're seeing mainly common variants in genes, polymorphisms as we call them, that increase risk for the disease but don't necessarily guarantee it. And the best established one is in a gene called ApoE, or apolipoprotein E on chromosome 19. And it comes in 3 major flavors. E3 is in 75% of the population, and with regard to Alzheimer's disease, it's neutral. But E4, or epsilon 4, as we call it, this is in 20% of the population. So 1 in 5 people in this room carry an epsilon 4 variant. But if you look at the Alzheimer's population, its prevalence goes up to 50%. So that drives a genetic association where ApoE4 is associated with increased risk for Alzheimer's. And then much work has been done on this over the years, such that we know if you carry 1 epsilon 4 allele, your risk for Alzheimer's is increased over 3-fold versus somebody who carries just the E3s. If you carry 2 E4 alleles, which is probably only 2 or 3% of the population, you have a greater than 10-fold risk for Alzheimer's. So this doesn't guarantee Alzheimer's disease, but increases risk. In contrast, if you have an E2 together with an E3, it's protective. So much research is going on now to understand how these different variants of ApoE affect its function. What does ApoE do? ApoE is involved with your cholesterol metabolism. It's a cholesterol carrier, and it's involved with delivering cholesterol to cells. If you have an E4, you're also at higher risk for heart disease. You're also at higher risk for high cholesterol. Unfortunately, when you ask, okay, I showed you earlier that high cholesterol was a risk factor for Alzheimer's, so you might think, okay, E4 increases your cholesterol levels, that causes risk for Alzheimer's. It doesn't work. When you actually look and you say, okay, let's look at the people who carried E4 and got Alzheimer's versus those that didn't, and what's the difference whether they had high cholesterol, it doesn't turn out to be statistically significant. So there's something else going on with ApoE beyond cholesterol. And it looks like it may involve the clearance of a beta peptide in the brain. So what we know then is that whether you have the early onset gene mutations that are rare and guarantee disease, or the susceptibility variant of ApoE, that in the end you have increased A beta accumulation in the brain. This appears to be via altered production of A beta, where you favor 42 versus 40, which drives more aggregation. And ApoE, the best bet, I don't have time to review the data of people like Zlokovic and Holtzman, but ApoE affects the clearance rate of A beta out of the brain. So here's a complicated slide that shows what we've learned so far from the 4 known Alzheimer's genes. Here's APP up here. Here's the A beta portion. You can either get what we call the good cut alpha secretase that clips A beta in 2 pieces, so you can't have intact A beta produced. Or if you get this cut, beta secretase followed by gamma secretase, you release A beta peptide. The mutations in APP and the presenilin 1 and 2 genes, and remember, presenilins are part of the gamma secretase clip here, they're the active portion of this enzyme. These mutations in APP and presenilins increase the ratio of A beta 42 to A beta 40. When you have a little bit too much 42 versus 40, this drives aggregation of A beta. We also learned back in the early 90s in my lab that aggregation of A beta is driven by the metals copper and zinc. In fact, if you take copper and zinc away from a solution of A beta altogether, you won't form any dimers or trimers or oligomers. It will stay as a single A beta peptide as a monomer. So this is a metal-driven reaction. As you get these aggregates, they accumulate, they can form within synapses and impair neurotransmission, or as we call it, long-term potentiation. Long-term potentiation is the process by which you consolidate synapses as you make new connections in the brain. Then eventually, these little aggregates will grow into big fibrils, and you get a plaque. This can drive oxidative stress and inflammation and nerve cell death. Somewhere in this process, you also form inside the neurons the neurofibrillary tangles, which are made up of a protein called tau, and this also drives nerve cell death. Now, the good pathway is that as you make A beta, the brain goes out of its way to clear it out. So if you have a single A beta made in the brain and it doesn't aggregate and it stays as a single entity, it is shipped out of the brain generally within 8 minutes. So it's one of the fastest molecules after being made to be shipped out of the brain within 8 minutes, according to the work of Berislav Zlokovic. So A beta gets out of the brain with the help of molecules like ApoE. And we know there's a difference in terms of how well E4, the risk factor, gets A beta out versus E3. E4 is not quite as effective, and this is one of the clues we have of how ApoE acts as a risk factor. A beta can also be degraded. It can be degraded after it leaves the brain in the bloodstream, or it can be degraded directly in the brain. And this arrow goes both ways, because after A beta gets out of the brain, it can make its way back in. It can also cycle back in. And all this information will be important later on when I give you the quiz on how we're going to cure Alzheimer's disease. And I'm not kidding. We're going to get there. Yeah.
You're associating with the aggregates with inflammation. Couldn't the oligomers
Yes.
also cause inflammation?
Yeah, I have inflammation down here being driven by the fibrils. And the question is, could the smaller aggregates, the oligomers, also drive inflammation? And yes, absolutely. It's just tough to get it into the slide, but yes. Yes? how would nutrition or diet affect these cuts? It appears that high cholesterol increases the production of A beta in the brain. And so although statins, Lipitor, have not been effective so far in treating Alzheimer's disease. And we're not quite sure how cholesterol does that. My wife's work, Dora Kovacs, she has worked at a particular cholesterol enzyme called ACAT1. ACAT1 is central to how high cholesterol drives A beta production. And she's testing now ACAT inhibitors for treating Alzheimer's disease. But other than that, unless somebody else in the audience knows, maybe Tuck has some ideas, not a whole lot about how diet affects these cuts other than cholesterol levels. Anything to add there, Tuck? Or?
Caloric restriction.
Yeah, thank you.
Chokes down amyloid neurodegeneration. We really don't know how.
Yeah, so caloric restriction, which is the only proven way, I think, to really stop aging in most organisms, it also has been shown. I think it was Mark Matson's paper. Was it Mark who showed that, or you showed that? Or?
We should.
You showed it, sorry. See, I always put my foot in my mouth, Tuck. So Tuck has shown that caloric restriction will also reduce A beta production, although the molecular mechanism, we're not sure how that happens yet.
Don't miss lunch.
Yeah. Exactly. And red wine is protective, so just drink wine with your dinner and exercise. Physical exercise is probably the best thing you can possibly do to beat Alzheimer's. So here's where we're at. We talked about the genetics of Alzheimer's, 4 known genes that are involved so far. But here's a big point. Only 30% of the variants of Alzheimer's is explained by those 4 genes. And if someday we want to carry out what I think would be the answer to eradicating Alzheimer's, a personalized medicine strategy, we need to identify all the genes. And what that means is that rather than waiting for Alzheimer's to strike, if we really want to attack this disease now and in the future, we need to prevent the disease before it strikes. The personalized medicine approach, also called pharmacogenetic approach, is based on early prediction. So you use all the genes identified to predict early who has the disease, make sure genetic privacy laws are in place, make sure genetic counseling is done properly. If one is at increased risk, early detection. The future use of biomarkers, like we discussed, the A beta in the bloodstream and in the cerebrospinal fluid, early detection to see who's showing like to have pre-symptomatic problems, maybe around 40, 50 years old. And then early prevention. Start either changing lifestyle early on in life or supplements or therapy, pharmaceuticals, whatever necessary to prevent the disease, but you need to do this before it strikes. So when I was in Washington a couple of weeks ago and presenting a national research strategy for how can we try to aim toward eradicating this disease by 2020, and we really pushed for this idea that let's find all the genes, let's learn from them in terms of how they cause the disease, think about drug strategies, and then eventually use this information to predict the disease early, use biomarkers and imaging to detect early, and then think about drug therapies to prevent this disease before it strikes. That this is our best bet for really getting rid of Alzheimer's. And I suggested that we increase funding for Alzheimer's to $5 billion per year, and nobody really raised any eyebrows that much, at least not while I was looking. But I think that's what we need to do. This is a huge problem, and it's going to completely crush this country in terms of health care and Medicare and Medicaid, and we need to think now for the next 10 years to make a big push. And I just think that this is one way to do it, and many other people believe as well, that let's prevent this disease before it strikes, and that means you need to predict who's at greatest risk. So how do we get there? We need to identify all of the AD genes, and I think there will be dozens, perhaps over 100, of variable effects, minor, moderate, and strong. Genetic counseling, psychological counseling. Important to have genetic privacy laws in place. This is still an ongoing process in this country. Detect the disease early, biomarkers, imaging, other strategies, and most importantly, start preventing this disease as soon as you know you're at increased risk. That can mean lifestyle changes starting at 10 years old. And if necessary, if that's not good enough, and it won't be in some people, pharmaceuticals starting at 40 or 50 if necessary. But I think it's going to be different for each person based on their genetics. So that's why this is called in the field personalized medicine. So how do we get there? And this is where I want to talk about the search for finding the other Alzheimer's genes beyond the ones we know about. And I think I could just summarize it. Since 1995, when the last of the 4 established Alzheimer's genes were found, there's been a huge amount of confusion and frustration about Alzheimer's genes. And like we used to say, well, finding a gene for Alzheimer's is like trying to find a needle in a haystack. But now the problem's a little different. There are too many people finding too many needles, and we don't know which ones are correct. Because in genetics, you can get a lot of false positive results, false negative results, depending on how good your sample is, how big it is, how your experiment was designed. So we really need to be careful on how we go over the data. So let me just illustrate this. There are 2 ways to do genetics. You can do what's called linkage analysis, where you look down through generations and big families and look at the segregation of a disease from generation to generation. For that, you need big families, and that worked really well with early onset Alzheimer's, where the families were big. You had lots of affecteds because, if they're getting the disease at 50 years old, they're not dying of other causes, so you can see big clusters of familial Alzheimer's. But in late onset Alzheimer's, which is the majority, you don't see the big families. So we do what's called association, where, like I showed you for ApoE, the E4 allele was in 20% of the general population, 50% of the cases. That means you have association of that E4 with the disease. So this is what's done with case control samples. You can also use family-based association. I won't get into the specifics here, but this is the main way that we've going forward in terms of finding the remaining Alzheimer's genes is association studies. So you're basically going to cross the entire population and asking for gene variants that, that are concentrated in the disease. The gene variants we look at are called single nucleotide polymorphisms, or SNPs. And what you need to know is that between you and the person sitting next to you, unless it's a family member, there are 3 million SNPs between you and anybody else. So if you have 3 billion bases of DNA, every basically million or so bases, you have a variant that's different in you versus the person next to you. It's what makes us all different. If you look at all of humankind, there are 30 million SNPs across all humans. That's a lot of variants, 30 million in 3 billion bases of DNA that we have in our genome. And some of these SNPs mean nothing. They're neutral. Some can cause early onset AD with certainty, like the early onset mutations. Some can increase your susceptibility, like E4. Some can modify the rate of progress of your disease. Others might determine whether you're going to respond to a certain therapy with safety or efficacy. It's these single nucleotide polymorphisms that we track, and we try to find association. The first single nucleotide polymorphism used was in 1983, and I was really lucky to be a part of this study with Jim Gisella, who was the first to test the first SNPs, which were called RIFLIPs back then. There was a set of 12. And I showed this slide because back then it took us 3 years to test 12 SNPs in one large family. And today, as I'll show you in these genome-wide screens we're doing, we can test 1 million SNPs in over 400 families in 6 months. And it's just really cool for me personally to have been, the hands. I wasn't the brains on that. Jim Gisella was the brains. But I was the hands on that project. And the very first time we used the first SNPs ever to find a disease gene and how long it took. And now to see, be here 25 years later and testing 1 million SNPs at a time in hundreds of families because of where the technology has taken us. And it's because of that advance in technology that we have our first shot to find all these other Alzheimer's genes and move forward. And we call this the Alzheimer's Genome Project, and I want to explain a little bit about that. So if you look across the literature, there are over 1,000 publications that have tested over 500 candidate genes, over 1,600 different SNPs for association with Alzheimer's disease. That's why I showed all the needles in the haystack. About 30% of these initially claim association with the disease, and then you start looking at the other papers that are published each month. There are about 10 Alzheimer's Association publications per month, but most of these positive associations are never replicated. Some of them are partially replicated. Only ApoE is replicated every time. ApoE is a clear-cut risk factor, and so we put ApoE like this. There's no doubt about that one. So how do we find the ones that are smaller? And we have a website for those aficionados, alzgene.org. And if you go to this website, we update it every few weeks. And I say we, it's Lars Bertram and I. Over 1,100 genetic studies are covered from 1978 to present, when, over 1,800 different SNPs and over 550 different genes. And most importantly, so like I said, when you get this initial association published, most of them no one ever replicates. But here's the thing. For many of these, you put the original paper comes out. They say, oh, we see association with this SNP in gene X. And then there'll be a slew of papers with different samples. And some will say, yes, we see it. And some will say, no, we don't. And in the old days, you just used to believe whoever your favorite geneticist was. So you say, well, Tanzi didn't see it, but Hardy did, so I'll believe Hardy. So, but we think that you can do this a little bit more objectively and statistically. So what we do is for every single SNP that's been tested in at least 4 independent samples, and that could be in the same paper, but if 4 independent case control samples have been tested, we run what's called the meta-analysis. We take all the data together. We have people dedicated to doing this. And for over 200 of the 1,800 SNPs that are covered, but over 200, they have been running over 4 independent samples. So we can carry out meta-analyses and say, of those 200, how many still show statistically significant evidence for association with disease when you compile all the data together, okay? And of those 200, 28 genes come up positive in Alzheimer's disease. And that's pretty good. So if you went to the website today, this is the front page. And if you clicked here on top results, this is kind of like ESPN Sports Center. You just get the scores at the end of the day. You don't have to watch the game. So if you go here, you will see, sorry, let me get this up. You will then see, okay, PowerPoint going wild. Let me. I thought I had a slide that blew this up, and I apologize because I guess I have it coming up later. But you would see the top genes here, and they're rank ordered. ApoE is number 1, and then they're rank ordered by size of effect. And I should make the point that some of these gene variants affect risk, but some of them also confer protection. That in this study, these types of studies, you don't just find risk factors, you also find SNPs that can confer protection. And someday, if you think about it, you're going to have all this information to calculate your cumulative risk. This slide was just to show that we're doing the same thing for schizophrenia, Alzheimer's, and Parkinson's disease, szgene.org, schizophrenia, alzgene.org for Alzheimer's, pdgene.org for Parkinson's. And what's cool is when you get the best hits from each one, you can now start to see commonalities. Like ApoE, which is number 1 in Alzheimer's, also turns up in the middle of the pack for schizophrenia and Parkinson's disease, which is kind of interesting. What does it mean? Only Tuck can figure that out. And then you have SNPs here that show up in both Alzheimer's and schizophrenia, Alzheimer's and Parkinson's, etc. So now you can start going across these collective analyses and looking for common genes across these different diseases. And we're going to be adding an autism and a multiple sclerosis version of this as well pretty soon. So here's the list. These are the 28 genes I talked about. ApoE is number 1. Number 2 is the nicotinic receptor, beta 2. That SNP is protective. And what's interesting is that there are several companies now developing drugs that turn on, that bind to this particular part of the nicotinic receptor, which is involved with learning and memory. And so I'm very, these are going to be, these will be symptomatic drugs, but it's set, at least the genetic supports that's a pretty good target. So I don't have time to go through all of these different genes, but if you go to the website, this is the list. But I do want to make one point. And I do this with fish. And I'm a vegetarian, but I have to use fish for this slide. That's a very big fish. You can see the guy back here holding it. These are the 4 known Alzheimer genes: APP, preselin-1, preselin-2, ApoE. They're big fish. And why I show this is because if you go back to this, so here's ApoE, and then if you go to the next 27 genes on the late onset risk factor slide after ApoE, they look like this. Okay? They're very little fish. But they are fish, and they can still feed you. And my point is that even though the genes that showed up so far through these case control studies on Als gene, where people pick their favorite gene and test it, And then some people replicate and some don't, and you do meta-analysis. Even though they're small, someday we're going to need to know all of them, all the small risk-conferring and protection-conferring variants to do proper genetic testing. So you want all the fish, big and small. So how do we find the bigger fish? And this is what I want to start to close with. And this is something we call genome-wide association screens. And what's interesting about this, and this is what I was talking about, where we can test 500,000 or 1 million SNPs at once on these chips using very new technology. This is unbiased genetics. Everything I showed you so far, somebody had a good idea. They said, I bet you this gene causes the disease, and they tested as a candidate, and they hoped for the best. And I showed you the 28 genes out of 550 that people have come up with that look promising, but they're all little fish compared to ApoE. This experiment, you don't pick the genes in advance. You screen the entire genome. You have SNPs, variants covering the whole genome. You test them in either thousands of cases and controls, or in our cases, we look at about 400 or 500 families, and you ask which ones show association, and then you ask, okay, this SNP shows association. By the way, what gene is it in? And then you get the gene. And this is the best way to do genetics. You do genetics unbiased, and you want to be surprised by the results. And we certainly have been surprised so far. We just published our top-line results of our genome-wide screen. Like I said, we tested in our first set of studies, we tested 500,000 SNPs in 437 families, 1,500 subjects. And then we put them through replication testing in 900 more families, 5,000 more subjects. So this was a huge, expensive experiment. And in the end, of course, ApoE came up. ApoE always comes up. But we also got 4 new genes for Alzheimer's. Time magazine actually named us a top 10 medical breakthrough for 2008, which helped a lot with fundraising and donations. And this just shows basically, I won't get into all of the study details because time is getting late. I'll just get you to the results. Beyond ApoE, these are the 4 genes we found. And one of them is on chromosome 14, near presenilin 1. But we have no idea what it does. Totally novel gene. We're starting from square one to figure out what it does. But that's where we were in 95 with presenilin. Before we all figured out that presenilin is part of the gamma secretase that clips APP. We had to start with just a new gene, so that's what we have again on 14. We have a gene I want to talk a little bit about called CD33, which is part of the innate immune system. This gene, SCA1, causes spinocerebellar ataxia. This is another neurodegenerative disease that affects movement, the cerebellum and the spinal cord. It's coming up in Alzheimer's disease. But it's not the same mutations that cause that disease. And finally, we have a gene here that's part of the synapse. At least this one made sense. This one is part of the synapse, which is involved in neurotransmission. But all of these, of course, are pretty big surprises, and all of them showed what's called genome-wide significance, meaning that they're coming in with very strong results. Ataxin-1 is the first one. This is the gene that causes spinocerebellar ataxia. It's a late-onset neurodegenerative disease. It's caused by an expansion of glutamine amino acids in this region here. And the pathology is characterized by progressive motor deterioration. So this is a movement disorder, the cerebellum being a movement region of the brain. And when we looked for this glutamine repeat expansion that causes ataxia, in the Alzheimer patients, it wasn't there. So we know that same gene, but there has to be different mutations. We're trying to find those mutations. In the meantime, we do have very strong data that somehow this gene affects the production of A-beta from APP. When we silence this gene with RNAi, turn off this gene, both A-beta 40 and 42 levels go up. If we overexpress the gene, we can drive the A-beta levels down. So gene activity up, A-beta down, gene activity down, A-beta up. And it looks like, without getting into the details here, that it's affecting that first clip of APP, the beta secretase clip. Somehow, when you turn this gene off, you get more beta secretase cleavage of APP. Totally unexpected. This is a disease that we never really associate with Alzheimer's disease, but the unbiased genetic experiment says, take a look at this. The next gene I want to spend some time on is CD33. This is a gene that's involved in the innate immune system. So for those of you who don't know, most of you know about the adaptive immune system, antibodies, T cells, etc. But the oldest immune system we still have, and the one the brain uses, because the brain doesn't have antibodies. The brain does not benefit from the adaptive immune system. Brain still does it the old-fashioned way, the way oysters do it and crabs and lower organisms. Basically, there's inflammation, there's glial cells, and most importantly, the innate immune system, which is your first defense against pathogenic bacteria, viruses, funguses, and yeast, is antimicrobial peptides. So the innate immune system allows you to make peptides. Same thing like A beta. The peptide comes from a larger precursor protein. So you have this big protein, you cut it in two places, you release a peptide, but in this case, the peptide is acting to kill bacteria, viruses, and yeast, so we call them AMPs, antimicrobial peptides. Well, this isn't the only gene we found in the innate immune system. This is the one we published because it had the strongest genetic statistics. But we had probably about a dozen different genes that came out of this genome screen that all kept hitting us in the head with the innate immune system. And Rob Moyer in my group started looking into the innate immune system and started asking, what if A beta peptide, which we think is a bad boy, is a good boy? What if A beta is an antimicrobial peptide? What if it means well? And I think back to, I ran a Gordon conference with John Tower, who's here today back, what was that, John, 10 years ago? Or it seems like 20 years ago. But Tuck Finch was our plenary speaker, and Tuck was making the point about the double-edged sword of inflammation, that the innate immune system, inflammation, it means well, but too much of it can cause bad things. So you don't want to just turn it off, but it's a system that is trying to protect the brain, but too much of it can cause, let's say, peripheral or friendly fire. You can start killing neurons if it gets too rambunctious. And I think that's what's happening here, but the new twist is that A beta peptide may actually be an antimicrobial peptide. I'm just going to cut to the chase here and show you all these different infectious organisms, Candida albicans, Listeria, Staph aureus, including the metallicin resistant, the drug-resistant type, E. physalis, Staphylococcus, Streptococcus, all your bad guys, okay? And then we compared it. We looked at the ability of A beta 42 and A beta 40 to stop the growth or kill these bacteria. And we compared it to the best known classic antimicrobial peptide in the innate immune system, LL37. Now in this graph, the MIC, the lower the number in this chart, the more potent the peptide is against that particular bacteria or yeast. And what you can see is that against Candida albicans, which is the yeast that grows in the mouth in thrush, and also you see it in immune-compromised individuals, A beta actually is much, much better than LL37 in killing Candida albicans. The level is 0.78 here versus 6.25. The lower number is better. A beta is also very good against Listeria, Staph aureus, E. physalis. And in some cases, A beta is better than the known antimicrobial peptide against some of these bugs. So it looks like a classic case of a new antimicrobial peptide in the innate immune system. But in this case, it's our old friend A beta from Alzheimer's disease. So this has got us thinking about different ways. Let me just show you just one piece of data. This just shows how A beta is bacteriostatic, meaning instead of killing the bacteria, initially it stops the growth of the bacteria, and then the bacteria fight back and eventually produce things that kill the peptide so the bacteria can grow again. So this just shows, for example, LL37 and A beta head-to-head. We also know that eventually A beta binds to the bacteria, these brown spots, the blue spots of bacteria. So this is just incip media alone. And the A beta actually binds and fibrillizes, actually forms oligomers and fibrils around the bacterial membrane, and not only stops their growth, but encapsulates them and allows them to cluster. And eventually, I don't have it here, but by electron microscopy, by EM, we see that the bacterial walls break open and the bacteria is eventually killed. This is exactly how it's done, for example, in crabs and in oysters. The peptide clusters, fibrillizes the pathogen, and then eventually the pathogen dies. So it looks like it's following the mechanism of an antimicrobial peptide. So this got us thinking now differently about Alzheimer's. There's been a literature that Alzheimer's could be a persistent subacute infection in the brain by pathogens such as chlamydia, Borrelia spirochetes, herpes virus, etc. We're now wondering whether A beta is being made for good reasons, with good intentions, that as we age, we're accumulating these pathogens, these microbes in our brain. A beta is being made to kill, is being produced to kill them. At Tuck's talk at the Gordon conference 10 years ago, Tuck mentioned about bacteria in the gingiva and how they may be infecting the heart. It's possible that, again, in cardiac amyloidosis, perhaps that amyloid is meant to kill those bugs. We've tested some other amyloids. We tested the amyloid in diabetes called amylin, which builds up in the pancreas. It also had antimicrobial effects. So this is creating a little paradigm shift, at least in our own minds, that maybe as we age and we start to accumulate these infections, even though they're not infections that will kill you, it's because the innate immune system is using misfolded proteins, amyloids, in order to kill these bugs. From a genetic standpoint, is it possible then that the Alzheimer genes are regulating the magnitude of one's innate immune response to microbial pathogens that infect our brain as we age? One of the things we know about Alzheimer's is that the first area affected is the entorhinal cortex feeding the hippocampus. Entorhinal cortex attaches to the olfactory bulb and through the nasal passage. So if you had constant bacterial or yeast or fungal or other infections from the nose, the first stop in the brain is exactly where Alzheimer's pathology hits. Past the olfactory bulb at the entorhinal cortex, feeding sensory information for short-term memory storage in the hippocampus. That's the area that's severed and where you always see amyloid. So we're thinking a whole lot differently because of this unbiased genetic experiment that perhaps infection with age through the nose, perhaps the gingiva, or a weakened blood-brain barrier, that over time the brain is fighting back by producing beta amyloid as an antimicrobial peptide. And so genetics is actually, the AD genes are actually dictating the robustness of your response in terms of how much amyloid should be made. So if you don't make enough amyloid, perhaps you will suffer from brain infection or meningitis. If you make too much, great. You kill the bugs or you kill perhaps the virus. We haven't tested herpes yet. We're testing that now. But at the same time, you're now at risk for Alzheimer's because you have too much amyloid produced, even if it was made for a good cause. I should also make the point that in AIDS dementia, that these patients have tons of beta amyloid in their brain. Nobody knew why. They have beta amyloid plaques. They also have amyloid inside of neurons. So we believe that this will go beyond the bacteria and yeast that we studied, and perhaps even viruses may play a role here. AIDS virus, we're looking at herpes virus, et cetera. So this just shows the kind of fun you can have, what can happen when you do an unbiased genetics experiment and taking you into new areas in terms of what's happening in Alzheimer's disease. So let me end then with just the quiz. We have someday we're hoping a chip that will contain all the different Alzheimer's genes on it, the early onset genes, ApoE, a few more big fish, lots and lots of little fish, perhaps ones to determine your drug response. But if you're going to test somebody for risk and they're at increased risk, you want to help them. And the drugs we have right now for treating this disease aren't very good. They're better than nothing, but basically they're drugs, they're cholinesterase inhibitors like Aricept and Exelon, Namenda, which hits a different neurotransmitter. But these drugs only hit the symptoms. They don't stop the disease. And in most cases, their benefit is temporary. So how do we get to drugs that will really work? And I show this because basically the roadmap we think about is just like we've been doing so far for the last 25 years. Discover the genes, understand how those genes cause the disease, take subsets of those genes that are amenable to drug discovery, screen for new drugs, and then take a subset of those that look viable for clinical trials. And so this is the pathway that we think we continue to need to follow. And so far, we've been limited to what we know about the original 4 genes in this pathway. So let me ask, this is what we have so far, symptomatic drugs. Everything goes wrong, neurons are dying, and then you try to treat the symptoms. So anybody want to just come up with an idea for other ways to treat the disease higher up this pathway? And anybody at all? Well, not Tuck.
Working on aging itself.
So aging itself.
So you know there is a lot of interventions in addition to calorie restriction to delay aging. Right? So what do you think the
So you,
for that is?
where will it hit? But aging will hit up here then, right? If you try a caloric restriction would prevent A beta production.
Dramatically, right? The whole system would be aging more slowly.
Well, yeah, I mean, hitting the whole system, aging more slowly.
And I mean, in some cases, for
Yeah.
example, with the dwarf mice, the cancer is not just delayed, but it's reduced. So the mice live longer, we reduce cancer. Is that, do you think, is it possible for Alzheimer's?
I'm not sure exactly how therapies that affect aging or lifespan are going to affect Alzheimer's. I think the good news is caloric restriction does, so there may be hope there. But I think you'd have to dive into the mechanism there, and I still think you have to pay attention, as I'm sure you agree, to the molecular details of how you get there. But yeah, I mean, yeah. Yes.
I'd concentrate on 2 areas. One, aggregation, and the other is
Okay, so those are on here. That would be, so let me just, let's just keep going through. So hold that thought. And any other ideas then? So we have prevent aggregation, promote degradation. How else do we fill in the chart? There's an obvious one up here. Prevent production. You can prevent tangles. You can also try to protect cells against inflammation, overly rambunctious inflammation. So if we go through the different levels, the first level of attack is try to prevent the production of A beta 42. And there are companies that are now trying to do this with what are called gamma secretase modulators. There are also companies that are trying to block A beta production in general with gamma secretase and beta secretase inhibitors. I'm a little afraid of those because I think A beta means well. I'm not sure I'd want to just block A beta production altogether, especially what we've learned about its potential role in the innate immune system. But you could think about modulating the A beta 42. And let me just fill these in. You can try to block aggregation here. The company I work with, Prana, has a drug that prevents the metal interaction with A beta. When you don't let copper and zinc interact with A beta, you prevent the oligomers from forming, and those drugs are looking very good. Drugs called PBT2 is looking very good in clinical trials, phase 2. For degradation, promoting degradation, which we heard suggested, there's A beta immunotherapy. And here it's a little bit more problematic. The idea here is you inject antibodies to A beta, and these antibodies are supposed to get into the brain and promote the degradation of A beta by microglia. It's also possible the antibodies bind A beta in the blood after A beta gets out and then sequesters A beta, doesn't let it back into the brain. Both mechanisms may be at play. Problem here is that, as you may have heard, Elon Wyeth, Wyeth was just bought by Pfizer, had the most advanced phase 3 trial going for an antibody called bapineuzumab, which is meant to do this, and they've had to now terminate the highest dose, which was the only dose that worked in phase 2, and only in patients who didn't have the ApoE4. So, and because the highest dose was causing inflammation in the brain, what's called vasogenic edema, the buildup of water around the blood-brain barrier, and in some cases microhemorrhage. And I think what's happening, and many others think this, is that most of the companies, and there are a dozen different companies, making these antibodies to A beta and injecting them, it's called passive immunization. These antibodies recognize the blood vessel amyloid I showed in the first slides. When those antibodies stick to blood vessel amyloid, two things go wrong. One is that the antibody can't get to its site of action, so it's held up, so it can't do its job. Second, as the antibodies stick to the amyloid in the blood vessels, it can cause inflammation, vasogenic edema, and if too far advanced, hemorrhage. So in my opinion, until a company does their homework and finds an antibody that will not see the blood vessel amyloid, but can still see the plaque or the oligomer amyloid in the brain, you're going to have these problems, the same problems that Elon Wyatt has. So Elon Wyatt now has had to go to only their low dose, which never even worked in the phase 2 trials. And I think that the immunotherapy is in a bit of trouble for that reason. Yeah. A specific pathogen?
Well, you think that the latest thing is that there are certain pathogens that and I was wondering if there's a specific pathogen.
Yeah. I mean, the front runners for the pathogens in Alzheimer's brain, we don't know which one's most relevant, but it's chlamydia, spirochete, and herpes virus. Well, they tend to, you would think would trigger the production of amyloid, so that so that the amyloid would presumably fight it. Yeah.
So isn't that in the target?
Yeah, I think so. I think yes. I mean, I didn't want to say it, but I mean, this is all pretty new, and I'm sure our paper is getting completely axed right now, where it's being reviewed, because anything new in Alzheimer's gets completely crushed. Yeah, I mean, we didn't go as far as to say that in our paper, but I would think that if you knew, and that's a relevant question, which pathogens are most likely with age driving the need for excessive beta amyloid and vaccinate against them, that would be great. I don't think it's going to be one pathogen. I think it will be multiple, but I think that is worth looking into, and that may be the future of preventing Alzheimer's, but it'd be, it's a ways off.
One other question.
Yeah, I'm confused about one thing. So you said that if you have antimicrobial response that drives the amyloid, and as a result of it, you wind up possibly doing some bad things. But earlier you said it's really the ratio of the matter, and so I don't see that connection.
Yeah, the connection is that to kill, I'm sorry, I should have made it clear. To kill those bacteria and yeast, you need a oligomer. You need A beta to aggregate. 42 is more effective than 40. If you increase the 42 to 40 ratio and mix it, it makes it more potent. And we showed the same thing now. It wasn't known for LL37, the known amp. We showed the same thing. We can drive its aggregation with metals. Nobody ever knew that, as we do with A beta. And as you drive oligomerization of LL37, you also can make it more potent. But 42, by driving oligomers, is more potent.
So in a test tube, can you do the enzymology to cleave the betas into the various sizes? It's not biochemically.
Yeah, it could be. And I think,
People doing
yes, Rob Moyer is now testing exactly which assemblies of A beta, different peptides, what they look like, and which ones kill which. And he thinks it's going to be like antibody diversity, that slight differences in different oligomers and their side chains will allow them to target different bacteria and yeast. But this is all very new, even in the innate immune system field.
We'd better close. Thank Dr. Tanzi very much, and
Thank you.
well, folks who signed up for lunch and have the name tag, you're cordially invited to have lunch in the courtyard.
That's good.
Really, that was a great